Alright everyone, let's talk about something super fundamental in our bodies: glycolysis. What is it? Simply put, glycolysis is the process of breaking down glucose, which is a type of sugar. Think of it as the first step your cells take to get energy from the food you eat, specifically from carbohydrates. The main goal here is to convert one molecule of glucose into 2 molecules of pyruvate. This whole process doesn't happen just anywhere. It specifically takes place in the cytoplasm of the cell. That's the jelly-like substance filling up the cell, outside of specialized compartments like the nucleus or mitochondria. It's a crucial pathway for life. Now, why is glycolysis so important? Well, it's one of the oldest and most widespread metabolic pathways known. This means it's found in nearly all organisms, from the simplest bacteria to complex humans like us. This universality tells us it evolved very early in the history of life. The energy it produces, though not massive in this single pathway, is vital. It provides a quick source of ATP, which is the main energy currency of the cell. Every cell in your body can use glycolysis to generate some energy, making it an absolutely essential process for survival and daily function, whether you're running a marathon or just thinking. The journey of glycolysis starts with glucose. Glucose is a simple sugar that we get from eating carbohydrates. Think bread, pasta, fruits. Once digested and absorbed into your bloodstream, glucose needs to get inside your cells to be used. Special proteins called glucose transporters, or GLUTs, act like tiny doors on the cell surface, helping glucose move from the blood into the cytoplasm. Once inside, the cell needs to trap the glucose, preventing it from leaking back out. This is where the first chemical step of glycolysis comes into play, a step we'll explore more soon. It's all about getting that sugar locked in and ready for processing. Glycolysis can be broadly divided into 2 main phases. The first phase is often called the energy investment phase. In these initial steps, the cell actually spends a bit of energy using up 2 ATP molecules. This might sound counterintuitive, spending energy to make energy, but it's like priming a pump. This investment sets up the glucose molecule to be split and eventually yield a net gain of energy. The second phase is the energy payoff phase. Here, the modified sugar molecules are further processed, leading to the production of 4 ATP molecules and 2 molecules of NADH, along with the 2 pyruvate molecules. So the net gain is 2 ATP and 2 NADH per glucose. The entire glycolytic pathway consists of 10 sequential enzyme-catalyzed reactions. It's a beautifully orchestrated assembly line within your cells. We don't need to memorize every single intermediate for a basic understanding, but knowing the key players and the overall flow is super important. The first 5 steps constitute that energy investment phase I mentioned earlier, preparing the glucose by adding phosphate groups and then splitting it into 2 smaller 3-carbon sugar phosphates. The subsequent 5 steps make up the energy payoff phase, where these 3-carbon units are converted into pyruvate, generating ATP and NADH in the process. It's a step-by-step transformation. Let's zoom in on the very first step, a critical one. Once glucose enters the cell, it's immediately phosphorylated. This means a phosphate group is added to it, forming glucose 6-phosphate or G6P. This reaction is catalyzed by an enzyme called hexokinase, or in the liver and pancreas, an enzyme called glucokinase. This phosphorylation step is super smart because G6P cannot easily cross the cell membrane, effectively trapping glucose inside the cell for further breakdown. Importantly, this step is irreversible under normal cellular conditions, meaning it commits glucose to the glycolytic pathway or other fates like glycogen synthesis. It's a one-way street for that glucose molecule. The third step is arguably the most important control point of glycolysis. This step is catalyzed by the enzyme phosphofructokinase 1 or PFK1. PFK1 adds a second phosphate group, this time to fructose 6-phosphate, which was formed from G6P in step 2, converting it into fructose 1,6-bisphosphate. This reaction is the major rate-limiting step of glycolysis. Think of PFK1 as the main traffic controller for the entire pathway. It's highly regulated, ensuring that glycolysis speeds up or slows down based on the cell's energy needs. Like the first step, this one is also irreversible, making it a key commitment point. Finally, let's jump to the 10th and final step of glycolysis. This step involves the enzyme pyruvate kinase. Here, phosphenylpyruvate, or PE, a high-energy intermediate, is converted into pyruvate. This reaction generates ATP. In fact, 2 molecules of ATP are produced at this step per original glucose molecule. since glucose was split into two 3-carbon units. This is a major part of the payoff. Pyruvate kinase is another one of the irreversible enzymes in glycolysis, and its activity is also carefully regulated to help control the flow through the pathway. So, we end up with pyruvate, ready for its next metabolic destination, and a net gain of energy. Why bother regulating glycolysis so tightly? Well, cells are incredibly efficient. They don't want to waste resources or produce energy when it's not needed. Regulation ensures that the rate of glycolysis matches the cell's actual demand for ATP. If the cell has plenty of energy, glycolysis slows down. If the cell is running low on energy, glycolysis speeds up. This careful control also helps to maintain stable levels of glucose in the blood and provide building blocks for other biosynthetic pathways when necessary. It's all about balance and responding to the cell's immediate environment and requirements. The regulation happens at the irreversible steps, primarily involving the enzymes hexokinase or glucokinase PFK1 and pyruvate kinase. Let's start with hexokinase. This enzyme, found in most tissues, is inhibited by its own product, glucose 6-phosphate. So if G6P levels build up because downstream steps are slow, hexokinase activity decreases. Glucokinase, found mainly in the liver and pancreatic beta cells, is different. It's not inhibited by G6P. Instead, its activity is indirectly stimulated by insulin. This means after a carbohydrate-rich meal, when insulin is high, the liver can effectively take up and process large amounts of glucose, which is super important for blood sugar control. PFK1 is the main star when it comes to glycolytic regulation. It's like the master switch. High levels of ATP, signaling that the cell has plenty of energy, act as an allosteric inhibitor of PFK1. This slows down glycolysis. Conversely, high levels of AMP, which indicate low energy status, act as an allosteric activator, speeding up PFK1 and thus glycolysis. Citrate, an intermediate of the citric acid cycle, also inhibits PFK1, signaling that alternative energy sources are plentiful. Perhaps the most potent activator of PFK1 in the liver is fructose 2,6-bisphosphate. The levels of F2,6BP are controlled by hormones like insulin and glucagon. Then we have pyruvate kinase, the enzyme at the final step. It's also subject to complex regulation. High levels of ATP act as an allosteric inhibitor, again signaling that energy is abundant. Interestingly, pyruvate kinase is allosterically activated by fructose 1,6-bisphosphate, the product of the PFK1 reaction. This is a cool example of feed-forward activation. An earlier intermediate in the pathway signals to a later enzyme to get ready for incoming substrates. In the liver, pyruvate kinase is also regulated by hormones. Glucagon, a hormone released during fasting, leads to phosphorylation and inactivation of liver. Pyruvate kinase, slowing glycolysis to conserve glucose. Insulin has the opposite effect. So why is all this biochemistry critical for our health and for medical students? Glycolysis is a primary source of ATP, the energy cells need to function. For some cells, it's especially crucial. Think about red blood cells. They lack mitochondria, the main powerhouses of other cells. Therefore, red blood cells rely entirely on anaerobic glycolysis for all their ATP needs. If glycolysis is impaired in RBCs, it can lead to serious problems. Also, during intense exercise, your muscle cells might not get enough oxygen to meet their energy demands through aerobic respiration alone. In these situations, muscles ramp up glycolysis to generate ATP quickly, even if it's less efficient overall. This brings us to some important clinical connections. In conditions of hypoxia, meaning low oxygen levels, like in poorly perfused tissues or at high altitudes, cells are forced to rely more heavily on anaerobic glycolysis for ATP production. While this provides some energy, the end product, pyruvate, gets converted to lactate. An accumulation of lactate can lead to lactic acidosis, a potentially dangerous condition. Understanding this is key for managing patients with shock or severe respiratory distress. It highlights how fundamental metabolic pathways can be disrupted in disease states, impacting patient outcomes significantly. Another fascinating clinical link is cancer metabolism. Many cancer cells exhibit a phenomenon known as the Warburg effect, or aerobic glycolysis. This means they preferentially use glycolysis for energy production, even when plenty of oxygen is available. They gobble up glucose at a high rate and produce lactate. This metabolic reprogramming is thought to support rapid cell proliferation by providing not just ATP, but also building blocks for new cells. This unique metabolic feature of cancer cells is an area of active research for diagnostics, like PET scans using a glucose analog and potential therapeutic targets. Also, inherited deficiencies in glycolytic enzymes like pyruvate kinase deficiency can cause chronic hemolytic anemia because RBCs can't make enough ATP to maintain their structure. Finally, for all you medical students gearing up for the USMLE or other board exams, glycolysis is high-yield stuff. You absolutely need to know the 3 main irreversible enzymes: hexokinase or glucokinase, PFK1, and pyruvate kinase. Remember that PFK1 is the rate-limiting enzyme, and its regulation is complex but crucial. Understand the hormonal regulation. Insulin generally stimulates glycolysis, think fed state, while glucagon generally inhibits it in the liver, think fasting state. Knowing these key facts about glycolysis isn't just for passing exams. It's foundational for understanding a wide range of diseases, from metabolic disorders to cancer, and for making informed clinical decisions. Stay curious.